Welcome back to another video tutorial from our lecture series on industrial microbiology. This is our third lecture on microbial production of industrial products, and the product that we are going to discuss today is the production of butanol. This has been quite a requested video, and hence today here I am to explain you the industrial production of butanol. So first let us see what is butanol and what are its uses. Butanol is also known as butyl alcohol, and I hope you all know what is the formula of butanol. It is C4H9OH. C4H9OH is the formula of butanol. Butanol has been used as a solvent for a wide variety of chemical and textile processes. It is also being used as a paint thinner and a solvent in other coating applications. Butanol has also been a component for hydraulic and brake fluids. It is being widely used as a base for our perfumes, deodorants, etc. And at last but not the least, butanol may also be used as a biofuel to reduce our environmental pollution. Next, we move on to the fermentation process of butanol, and the first step that we will be considering is the medium that is used for its production. So, first are molasses. Molasses are used as a raw material for the production of butanol. And let me tell you one thing, the raw materials that are used for the production of butanol requires some kind of processing before it can act as a raw material. So molasses, they require dilution and removal of sediments in order to remove any kind of minerals, because minerals might create any hindrance in the production process. Hence, it needs to be removed before it can be used as a raw material. Whey permeate is also used as a raw material for the production of butanol, and it requires concentration by reverse osmosis. For butanol production, corn also acts as a raw material. We have seen in our last two video lectures, which was in the production of ethanol and citric acid, corn has been a constant raw material for these products production, right? So in butanol, corn is used as a raw material, and the processing that corn needs to be an eligible candidate as a raw material for the production of butanol is it requires debranding, degerming, de-oiling, milling, sieving, centrifugation and saccharification. Okay, so debranding is the removal of bran from our corn. Degerming is the removal of the germs or the small seeds from our corn. De-oiling is removal of oils from corn. Milling is chopping it into small pieces, as we have seen that corn is being milled during the production of ethanol. Sieving is passing the corn through tiny mashes so that the fine particles get separated from the bigger particles. Centrifugation is separating the corn aqueous phase, and saccharification, I hope every one of you remember what is saccharification. We have discussed about it when we were studying the production of ethanol. Saccharification is the breakdown of starch into simple reducing sugars. Okay, and why do we need saccharification? Because corn doesn't have amylase, hence they cannot break down starch into simpler compounds. Okay, so we studied about the medium used and their processing. Now we will move on to the microorganisms that are used for the production of butanol, and you all will be very lucky because there is only one microorganism that is widely used for the production of butanol, which is the Clostridium. Clostridium acetobutylicum and Clostridium beijurnckii. These two microorganisms are used for the production of butanol, and with advancing time, Clostridium beijurnckii is the widely used microorganism for our butanol production. Why Clostridium beijurnckii? Like, what qualities does it have that it became the widest used microorganism for butanol production? So, Clostridium beijurnckii, it can tolerate higher concentration of butanol. We have studied during the production of ethanol that when yeasts are being used as the inoculum for production of ethanol, high concentration of ethanol inhibits the growth of yeast, which directly affects the quantity of ethanol that is being produced. In production of butanol, there might be some scenarios where the microorganism was getting inhibited due to high concentration of butanol, and hence researchers wanted some tolerant microorganism which can thrive through high concentration of butanol and still can produce higher yield of butanol. Hence, Clostridium beijurnckii won the race, and now it is being widely used. So, it can tolerate higher concentration of butanol, and hence it gives higher yields of butanol. Clostridium beijurnckii, it starts producing butanol during its active growth phase. When it is actively dividing, when it is at its active growth phase, it starts producing butanol, hence resulting in improved productivity. Means the quality, the quantity will be much higher. Okay. So, now we finally come to the process by which butanol is produced. So, the first point in butanol production is the type of fermentation. Okay. So, butanol is produced by batch fermentation process. Okay. It is produced by batch fermentation process. Another point that you have to keep in mind is that butanol production is also known as ABE production. Can anyone tell me the full form of ABE? ABE? If any one of you know the full form of AB, please comment down in the comment section below. In the meantime, I'll tell you, AB is basically acetone, butanol, ethanol. Okay? Butanol production is also known as acetone butanol ethanol production, because in earlier days when butanol has to be produced at an industrial level, acetone and ethanol were also being produced in some amount, and our researchers, those who have worked in this area, they have calculated, and today we know that acetone, butanol and ethanol is produced during the butanol production in a ratio of 3:6:1. That is, acetone is produced in 3 parts, butanol is produced in 6 parts, and acetone and ethanol is produced in 1 part. Let me explain it more clearly with an example. Suppose we are producing 10 ml of butanol. Okay. So during our fermentation process, we know ABE will be produced. They are produced in what ratio? 3:6:1. Hence, when we are producing 10 ml, 3 ml will be acetone, 6 ml will be butanol, and 1 ml will be ethanol. Is it clear? So, let's move on to the process of butanol production. And here I have summarized the entire production in a chart form, which will make it easier for you to understand that how butanol is produced. So in the first step we see is the corn milling section. In this vessel, corn and water is mixed, where corn is being milled, sieved, debraned, de-oiled. Okay? After which fiber, protein and germs, they are separated out. Okay? So the vessel now only contains corn syrup. What it contains? Corn syrup. Okay? The next step is what? Saccharification. Saccharification is the breakdown of starch into reducing sugars, into simple sugars. After saccharification, this fermentation broth enters into this fermentation section, which contains the inoculum, that is the spores of Clostridium beijurnckii, CSL. CSL is the medium. The full form for CSL is corn step liqueur. Okay? Corn step liqueur. So this fermentation vessel contains inoculum, corn step liqueur and some nutrients. Here, when fermentation is taking place, the cells are being moved out of the vessel. Here you can see the cells have moved out, as well as the gases which are being produced is being moving out of the vessel. This fermentation broth now moves to the distillation unit, which is known as ABE stripping distillation unit, that is acetone, butanol, ethanol stripping distillation column. Here what happens is acetone, butanol and ethanol will be separated by distillation process. Okay, it will be separated by distillation process. The fermentation broth contains some dissolved solids which will now move out of the vessel. Okay, the dissolved solid, suppose the cells, the medium, they will move out of the vessel, and ABE will get separated by distillation. Now from the distillation column, ABE enters the ABE separation column. Okay, it enters separation column. Here acetone, butanol and ethanol will be separated. So from here, from separation column, acetone and ethanol will move to this vessel and butanol will come to its butanol separating column. So acetone and ethanol moves to the acetone ethanol separation column and butanol moves to the butanol separation column. From here, the water which is left in the fermentation broth is removed out, which will be recycled for our next cycle of fermentation. The butanol which is separated out will be isolated. Butanol will be recovered from the separation column and acetone and ethanol will be recovered from this column. Hence, we come to the end of our today's lecture. I hope I have been able to make you understand the industrial production of butanol. If you like this video, please share it with your friends, with your family. If you find it very useful, please like our video and subscribe to our channel Scientistracademy. Thank you. Have a nice day.